Today we're going to be talking about cell respiration.
So the first question is, what is cell respiration?
Cell respiration is a process that releases energy by breaking down sugars in the presence of oxygen. It takes place in the mitochondria. The process of cell respiration produces 36 molecules of ATP, which is a form of energy.
Part of cell respiration is in a part of aerobic respiration. What aerobic means is that it requires oxygen. In cell respiration, oxygen balances out the electron concentration in the mitochondria and serves as the final electron acceptor in the electron transport chain. Without oxygen, cell respiration cannot continue.
Cell respiration can be put into a simple equation. The equation says that C6H12O6 plus 6O2 yields 6CO2 plus 6H2O plus energy. What this means is glucose plus oxygen yields carbon dioxide plus water and ATP, which is energy. Cell respiration can be broken down into three simple steps: glycolysis, the Krebs cycle, and the electron transport chain. And glycolysis literally means sugar breaking. In the Krebs cycle, it produces key materials to be used in the electron transport chain. In the electron transport chain, it mainly produces ATP.
The first step of cell respiration is glycolysis. To start the reaction, two ATP are invested. Then, the 1,6-carbon molecule of glucose is broken down into two smaller 3-carbon molecules. This molecule is called pyruvic acid. At the end of glycolysis, there's what's called the energy payoff. Since two ATP were invested and there was a gain of 4 total, there's a net gain of 2 ATP molecules. Also, two NAD pluses are given high energy electrons to convert to NADH. And then there are also the two pyruvates, which were created from the 6-carbon molecule of glucose. Now, before the Krebs cycle takes place, there's an intermediate step. The two 3-carbon pyruvates that just came from the glycolysis are going to enter what's part of the mitochondria that's called the matrix. Each pyruvate is going to give off one of their carbons that are going to be given off in the form of 2 molecules of carbon dioxide. Now, there are two 2-carbon molecules, and they're going to bind to what is called coenzyme A, which can be abbreviated to CoA. So now we have a new material called acetyl-CoA. Now, those 2 molecules of acetyl-CoA are going to enter the next step of cell respiration, the Krebs cycle, where more reactions are going to take place.
In the Krebs cycle, there are 3 reactants: pyruvic acid, NAD+, and FAD. Pyruvic acid is converted to citric acid, and NAD+ is converted to 6-NADH, which is like an electron carrier bus. FAD is converted to 2-FADH, which is also an electron carrier bus. In the process, 2 ATP is formed, and 4 CO2 molecules are released out of the cell.
So these products of the Krebs cycle will now go to the ETC, or the electron transport chain, besides ATP and CO2. Like Pat said earlier, the NADH and FADH are like electron buses. So what they do is they take the electrons to protein pumps within the membrane of the mitochondria or the electron transport chain. NADH has more energy, so it can take the electrons to maybe 2 or 3 more pumps, but FADH can only take it to 1 or 2 pumps. So what do the electrons do when they get to the pumps? Well, once NADH and FADH drop the electrons off, they go back to the Krebs cycle and leave the electrons there. But what the electrons do is they go down an electron transport chain to the ATP synthase. As the electrons are passed down the chain, hydrogen is pumped from the matrix into the intermembrane space. This creates a high concentration of hydrogen ions. So with this high concentration of hydrogen ions, they want and need to get back to the matrix to balance it out. So they will escape through the ATP synthase, which will create ATP, which we'll look into furthermore later.
Let's take a closer look at the ATP synthase. As you learned earlier, when the high energy electrons are moving through the protein pumps, they pump in hydrogen ions into the inner membrane space. Those hydrogen ions want to even out the concentration, so they are forced to move through the ATP synthase, which sort of turns a turbine, which adds a phosphate group onto ADP, which makes ATP, and ATP is a form of energy.
So now we have all these ATP molecules, and what exactly can they be used for? They can be used for energy anywhere in the cell. The energy can be used to perform different functions within the cell and to help maintain homeostasis.
So what is anaerobic respiration? Anaerobic respiration is a type of respiration where no oxygen is present. So during anaerobic respiration, O2 is not present, so H2O cannot be made. H2O will not be made in the ETC, which we talked about earlier. Without the H2O being made, it can't take away low energy electrons that make room for the high energy electrons that are being passed down the chain. This backs up the whole process all the way to glycolysis, which now has to transfer to fermentation.
Now the overall process of fermentation is actually quite simple. When oxygen isn't present, fermentation is going to follow the process of glycolysis. Now the high energy electrons that are from NADH are going to be passed back and donated to the pyruvic acid that is produced in glycolysis. Now when these electrons are dropped off, they're going to create one of two types of material. It's either going to create lactic acid or alcohol, and depending on which is created is going to dictate what type of fermentation it is.
Okay, so one type of fermentation is alcoholic fermentation. So what happens during alcoholic fermentation is NADH cannot drop off its electrons anywhere, so it donates them to the two pyruvic acids. So that creates an NAD+. What the NAD+ does is it's recycled back to make NADH again, and this process keeps continuing, and it makes ATP as it's going on. So the difference between an alcoholic fermentation and a lactic acid fermentation, which we'll talk about later, is the alcoholic fermentation produces CO2, where the lactic acid does not.
Now, like we said, the second type of fermentation is lactic acid fermentation. And the exact same thing happens where the high energy electrons are donated to the pyruvates, which frees up extra NAD pluses to transport the high energy electrons. Now, like we said, the difference between alcoholic and lactic acid fermentation is alcohol gives off carbon dioxide along with the alcohol, where lactic acid only gives off lactic acid. This is the fermentation that occurs in human muscle cells and different parts inside the human body as well.
So the equation for the alcoholic fermentation is pyruvic acid plus NADH yields alcohol plus carbon dioxide and NAD+.
And the equation for lactic acid is the same thing, pyruvic acid plus NADH, however the yields are different. They yield lactic acid and NAD+. There's no carbon dioxide yielded in lactic acid fermentation. And that's how cell respiration works. Thanks for watching. I hope you enjoyed it, and have a great day.